Okay. I hope my screen is visible. So, good evening, everyone. Thank you so much for joining today. Today, we are having the last interaction session for this course. First of all, I would like to thank everybody who has joined the sessions or gone through the video lectures, which are available on YouTube, and have benefited in any way from all of the materials that were shared either by me or by any of the other TAs for this course. I hope that it brought you some insight into the way research is being carried out currently in the field of cell biology, and it has somehow cultivated your scientific temperament and has made you a little more inclined to go into research. So, in that way, thank you so much for joining, and I hope this course at large was useful for you. Now, keeping that in mind, today will be mainly a Q&A kind of a session if anybody has any questions. And apart from that, I will also share some of the previous year questions for all of the weeks, which were uh, previously circulated as part of the assignments, because that will help you brush up everything that was covered as part of the course. And we'll also, if time permits, we'll briefly get a recap of some of the topics that we have covered in the live interaction sessions as well. So that is overall the plan. Apart from that, if anybody has any questions that you wish to take up before we get started, then please do. And then I'll get into the other questions. So if anybody has any questions, please mention those. Or if you want any topic clarified or something that you feel that would be a little more detail or some experimental support or some reference books that you might need, anything that might be useful, you can mention that. I'll reshare my screen with the questions in the time being. Okay, if not, then I'll start off with some questions. So, as part of week one, we had looked at some very preliminary topics about introduction to cell biology, the components of a cell, what are the different organelles, how do they work, what is the specification of or the prerequisite for each cell to be a eukaryotic or a prokaryotic cell, how is the cell organized, what are the basic processes. Then we looked at DNA as a genetic material. We understood how it works. We understood its necessity and the way it has evolved over time. We understood the chemistry of DNA and how it has till date persisted as the genetic material within the cell. So again, at any point of time, if there is anything you wish that you want to clarify, please do interrupt and we'll come to that topic immediately. It does not have to be even related to what we are discussing at the moment. So please interrupt me if there are any questions. Okay, so the first question is about the modern cell theory. So the modern cell theory does not include which of these following statements. First, that all organisms are made up of one or more cells. Cells carry hereditary information that is passed on to daughter cells during cell division. The cell is the fundamental and structural unit of living beings which have similar chemical composition and organization, or cells formed by free cell formation, similar to the formation of crystals. So, if you remember, the idea of or the concept of cell theory was something we had discussed during this week itself. How the initial observation of cells by Robert Hooke, then how Virchow came up with the theory, Schleiden and Schwann, they elaborated on the cell theory, and then what today, as science has evolved, as we have more and more information about cells, what is the current update or the current perception of the cell theory? So, among the options that are given, of course, the first option is correct, that organisms are made up of cells, and omnis cellula e celluli was the concept that was shared by scientists at that point of time, that from one cell you get more cells and those cells eventually constitute an organism. So the first statement is of course true. Secondly, yes, they do carry information or heritable information or the genetic material that gets passed from one generation into the next. And that is occurring during the cell cycle at the after the S phase, when cells are dividing into two, it is occurring during cell division. It is indeed this fundamental and structural unit of life, and therefore each cell is overall has a similar chemical composition and organization as well. Therefore, among these, the fourth option is the incorrect option. I think I had discussed this before, that even if there are certain questions where you do not recall the exact correct answer to it, you can always opt for answering MCQ questions by going through the options and going via the the process of elimination and arrive at the most suitable answer, which is most acceptable by what is currently known, and taking into consideration what was known at the time of discovery. So by the process of elimination also, you can come directly at the concept that is given here. Second question is showing you a bond between two different types of molecules, two types of molecules, which are both water molecules at the end of the day. But what you see is an interaction between the O atom, oxygen atom, and the hydrogen atom here. Now, basically, the question is asking for the type of interactions that are persisting here. There is one, the blue arrow here shows, I don't know, my pointer might not be working. But yeah, here you see one arrow indicated as A between O and H, and yet there is another arrow which is between O and H. So what we see between O and H from the same atom and O and H between different atoms. So the question that might come up is, why is there a difference at all? It is because of the nature in which these bonds have been formed and the way in which the atoms are interacting with each other. So here, the nature of the interaction or the bond between the intramolecular O and H connection or O and H interaction is covalent in nature, which is why you see it is indicated as a solid gray colored line. And the other one, which is indicated as which is indicated in a dotted manner, is a hydrogen bond, which basically indicates the nature in which hydrogen bonds are formed, that you see intermolecular hydrogen bond formation. So that was the correct answer to this. Then we are asked about activated carrier molecules. How do they store energy? So we have discussed about energy currencies within the cell, and we now understand it very well that it is indeed ATP, which is considered to be the energy carrier within the cell. But there are also other molecules like FADH2 or NADPH. All of these are considered to be energy carrier molecules or activated carrier molecules. The way in which such molecules are believed to carry energy or contain energy within themselves is via the bonds which they possess. Now, when we talk about bonds, the strongest bonds will persist between the atoms which are sharing electrons among each other, which means that they are involved in covalent interactions with each other, or they're forming covalent bonds with each other, because that has the highest energy. And therefore, when such an interaction is disrupted, the amount of energy which is released as a result of that disruption is the highest. And therefore, the correct option is option 1, that is the energy rich covalent bonds. In comparison, the other bonds, hydrogen ionic or hydrophobic, they have less energy, or the breakage of such bonds will release lesser amount of energy. Uh, then in question 4, we are asked which of the following modes of mobility of membrane phospholipids is the most energy consuming. So we had discussed the lipid bilayer or the plasma membrane and the nature in which the lipids are arranged with respect to each other, and the extent to which movement is allowed within these lipids. So we had discussed that if they are, when you consider the lipid bilayer, there are two distinct leaflets, the inner and the outer leaflet. The inner leaflet faces the cytoplasm, while the outer leaflet faces the surrounding environment, the extracellular environment. So movement of lipids among the or along the same leaflet is allowed, which means that it is considered to be lateral diffusion, that the lipids are only moving along the same plane, or they are moving laterally, and therefore it is a very low, relatively low energy consuming process. Or its rotation along the same place, or its flexion is also allowed, because those are less energy consuming processes. But the switch from one leaflet into another is made to be a very energy consuming process, because it can have far reaching consequences. If you remember, we had discussed about apoptosis and necrosis in one of the later sessions, and the point of distinction among, apart from many other points as well, one of the major points of distinction that exists for apoptosis is the fact that apoptosis is indicated, apart from other signals from within the cell, it is indicated by an eat me signal, which literally means that the cell is signaling to the external environment that it is now undergoing a programmed cell death process. For that, it allows the switching or the movement or diffusion, or just a switch basically, from the inner leaflet to the outer leaflet of one particular membrane phospholipid, that is phosphatidylserine. So phosphatidylserine under normal circumstances for a healthy cell is localized towards the inner leaflet, but when a cell is dying, its movement is allowed towards the outer layer, and that signals as an apoptotic signal and calls the surrounding, or it creates a microenvironment where the cell can easily undergo the process of programmed cell death. So because this is just one example of critical control that is executed, therefore from here we can understand that the flip-flop movement is something that is maintained very carefully and is therefore a very energy consuming process. Let me again please iterate that if anybody has any doubts related or unrelated to what we are discussing at the moment, please do interrupt. My camera is not working today, which is why it's not switched on, but I hope you can understand me. If not, please let me know that as well, and I'll try to repeat more clearly. Okay. Um, Coming to the next question, we've been asked to pick the correct option. So ion transporters actively move ions against the concentration gradient. Ion channels don't allow the ions to diffuse down the concentration gradient. Ion channels create ion concentration gradients, or ion transporters cause selective permeability to ions. So we have to understand what is the nature and the utility of ion transporters. When we talk about movement or transporters, they are of many different types. I think we have discussed it as well. You can either have passive transport, you can have active transport, or you can have uh, facilitated transport. So when it is passive, it means there is no energy investment. Anything can move from either side of the cell to the other side, whether it is from the cell interior to outside or vice versa. In contrast, when it is an active form of transport, their energy investment is required, and therefore it is an expensive process or an active process. In contrast, when you talk about facilitated diffusion, it means that there is some other compound which is facilitating or is allowing or is supporting this movement. Now, that is usually mediated by a bidirectional form of transport, where it also brings us to another concept of symport, antiport, etc. Symport means when um, two components or two particles or two, let's say, atoms or proteins, whatever it is, they are moving in the same direction. Antiport means that they are moving in opposite directions. So basically, when facilitated diffusion is carried out, many a times import or antiport of another molecule is also used. For example, there is an Na plus glucose transporter. So such transporters will transport Na plus or sodium, but they will also simultaneously carry out the transport of glucose at the same point of time. So it is co-regulating the concentration of two different molecules. These are the different subtypes. Among the options that have been given here, when we talk about the nature of ion transporters, they will, because they are transporters and because they are not, just passively allowing something to move from one direction to another, they will carry out this process of movement of ions in a very regulated manner. What they do, as the name indicates, transporter, since they are utilizing energy, they will do so against the concentration gradient. So the basic principle of concentrations that will obviously indicate the fact that if something is present in excess or at a higher concentration on one side, then the default direction of movement is from the direction of high to low. So anything that is usually present in excess on the other side, if it is allowed to passively move without any investment of energy, it will shift towards the side with a lower concentration gradient. In contrast, if you need to move that against this concentration gradient direction, then energy would be required. Therefore, ion transporters actively, which means that with an investment of energy, will move ions against this gradient. So that is the correct option. The others are, of course, not correct because A, channel will allow ions to move down the concentration gradient. Therefore, the second statement is not true. Ion channel creates ion concentrations gradients. No, like I said, channels will allow for passive diffusion. Therefore, they will always try to equalize the concentration on either side of the membrane, and therefore they do not support the creation of concentration gradients. And ion transporters cause selective permeability to ions. This is also not true. If an ion transporter is specific for one particular ion, it will carry out its transport irrespective of any other things. So therefore option 1 is correct. Then we come to Shargav's rules. So Shargav had stated, if you remember, we had discussed this during the structure of DNA. Then it basically indicates that the number of A is equal to the number of, if you just need to write it down, I think I said this as well, that A double bonds to T and G triple bonds to C. Therefore, the concentration or the ratio at which adenine is present should always be equal to the amount or the ratio that thymine is present. And for G and C, or guanine and cytosine, we see a similar distribution or similar prevalence, because A needs to double bond, or A needs to form a bond with T, and G needs to form a bond with C in order to create that double helical structure where there is complementarity between the bases. So taking this or keeping this in mind, what we can conclude is that the ratio of A to T and G to C will be close to 1, which means that they would be equal in number. And that is exactly what Shargav's rule says as well. Therefore, the correct option in this case is option 3, that A and T would be, the ratio of A and T would be equal to, would be close to unity, not equal, but would be close to unity. And similarly for G and C as well. So with this, we come to question 7. What is the difference between ribose and deoxyribose sugar? So obviously the distinction between them has a huge impact on the way in which DNA and RNA can be perceived. So basically the deoxyribose sugar, if you remember, we had the structure on hand. It contains hydrogen at the C2 prime position, whereas ribose had the OH group. The absence of this OH group, or um, the deoxy condition of the deoxyribose sugar, is what makes it different and gives rise to the name of deoxyribonucleic acid, or DNA. So therefore, the point of difference is the presence of OH group and H group between these two, and this difference occurs at the C2 prime position. So therefore, the correct option is option 1, which is for deoxyribose sugar having hydrogen at C2 prime position, while ribose has hydroxyl group at that same position. For question 8, we are asked what is the direction of the phosphodiester linkage that is formed between two nucleotides. So phosphodiester linkage, which we saw, is the backbone of DNA that occurs when one nucleotide triphosphate initially arrives, and then the subsequent nucleotide triphosphate, or NTP, DNTP in particular, is able to bind or form a phosphodiester bond with the previous nucleic acid or previous nucleotide, and then this continues till a polynucleotide is synthesized. So for this bond to be formed, the direction in which this fusion occurs, or this phosphodiester linkage occurs, is that the C3 of the first pentose will fuse with the C5 prime of the following pentose. So this was a direct question. This you need to keep in mind, and you need to use the basic logic in which nucleotide by polynucleotide or DNA formation exactly occurs, or DNA synthesis actually occurs. So that should make things clear for you. So that is the correct option is this first option, which is C3 prime and C5 prime of the two subsequent pentose sugars. For question 9, we have been asked actinomycin D inhibits which of the following? DNA-directed RNA synthesis, RNA-directed DNA synthesis, RNA-directed protein synthesis, or cDNA-directed protein synthesis. So actinomycin D is for inhibition of DNA-directed RNA synthesis, or for the process of transcription. So this is something we not much to elaborate on. That is the correct answer for this question. And finally, for question 10, if we have to look at the bonds that are present, we have just discussed this as part of the Shargav's rule as well, that 2 hydrogen bonds are present between A and T, whereas 3 hydrogen bonds will be present between G and C. So option 4 is the correct option for the B form of DNA. So with this, we come to an end for the questions for week 1. Uh, any doubts regarding this? Please to let me know. Okay. So if there are, again, interrupt me at any point of time. No worries about that. So, for the second week, the topics were centered around cell cycle, its regulation, its control, the different interactions between majorly the cyclin CDK complexes, the way in which their levels increase and decrease depending upon what is the external stimulus, what is the condition, whether the cell is starving or healthy, or what exactly is the need at that moment. And the way in which the cell cycle progresses or proceeds is also something that was heavily controlled by the cyclin CDK complexes with help from many other proteins also as well. So, majorly we looked at that, and also we saw the DNA damage, DNA replication checkpoints as well to gain further understanding of the same topics. So, first question is about the events of a mitotic cell cycle. This we have discussed like hundreds of times. So, the correct order, if you are already not familiar with it, then cells initially, when they are in the quiescent state, they are said to be in G0. From G0, they go into they go into G1 phase, S phase, G2 phase, and M phase. So there is G1, S, G2, M, and then an interphase until the cell undergoes division again. Therefore, that is the correct option, option 1. Second, we are being asked that the protein P53, which we have extensively discussed, and which is commonly also known as the guardian of the genome, induces the expression of P21 after checkpoint activation. So P53 controls P21 expression, and this occurs after checkpoint has been activated. What is P21? P21 we have is something we have discussed extensively as well. It is a CKI. So after P53 has induced P21, it is used to keep a check on the following cyclin CDK complexes, and it regulates this job via its CKI nature. Uh, then coming to question 3. In an experiment, when the cytoplasm from an M phase oocyte is microinjected into a G2 phase oocyte, what will be the possible effect? So, if you remember, we had discussed these cell fusion experiments that are quite extensively carried out. And in this, you are basically fusing the protoplasm, or rather the cytoplasm and the nucleus of different of cells that are currently in different stages or different phases of the cell cycle. So basically what happens is that when you are fusing a G2 phase, you are taking a G2 phase oocyte and you are adding the cytoplasm from the M phase oocyte. It means that the M phase oocyte is currently carrying the cyclin and CDKs which are necessary for progression through the M phase. As a result, the G2 phase oocyte, while it was not previously acquainted or previously set up for a process of cell division, it will now undergo the same because the presence of the M phase, M CDK and M cyclin, will push it to continue into division. Therefore, cell fusion experiments, they showed that there is a directionality in which cell cycle events continue, and that directionality is majorly being maintained by the cyclin CDK complexes. So the correct option is that the G2 oocyte will be driven or will be pushed towards the M phase. That is option 3. Then for question 4, we've been asked which of the following is the longest phase in the cell cycle. So we just discussed it right now. While the options here might look similar to you, they are not. Keep in mind that prophase, anaphase, and cytokinesis are all parts of the M phase, whereas the interphase is the duration or the period between two subsequent cell divisions. So the interphase is actually the most prolonged, or is the longest phase within the cell cycle, and it is required in order to ensure that there is no haphazard or incorrect or unnecessary proliferation of cells in any random location within the body. Therefore, interphases maintaining that control or non-dividing state of those cells. Then in question 5, we have been asked which of these are the incorrect statements. CDC 28, it is about CDC 28. So CDC 28 mutants, which of these following statements can be seen to be untrue for them or false for them? So A, do they not initiate DNA synthesis at all? Do they not progress beyond the start point? If they encode for a cyclin-dependent kinase, or can they progress into S phase? So the incorrect statement in case of CDC 28 operates at the checkpoint or the restriction point, and it makes sure that if there is any a disability, or if there is any sort of any problem or any undesirable situation for cells at any point of time, then such a cell will be restricted in the pre-synthesis phase and will not be allowed to proceed or will not be allowed to enter into the S phase. Therefore, the incorrect statement is option 4, that CDC mutant cells can progress into S phase. Yeah. Yeah. Um, Select the incorrect statement about CDC2 protein. So CDC2, which of these following are correct? CDC2 levels do not change during mitosis. Uh, CDC2 becomes phosphorylated on entry into the stationary phase. This statement is of course not true. CDC2 phosphorylation correlates with high mitotic index, or CDC2 has kinase activity. So the rest of the options are correct. The incorrect option in this case about CDC2 is that upon entry into the stationary phase, it becomes phosphorylated. And that will of course be the correct option. Which of the following enzymes is inhibited by hydroxyurea? I think this is also something we had extensively discussed during the live interaction session. So hydroxyurea is acting upon ribonucleotide reductase and is inhibiting the same. So that is why it is getting stuck, and the other options in this case are of course not correct. So the correct answer is ribonucleotide reductase, option 2. CDK activity is being regulated by a cyclin association, by the inhibitory phosphorylation in threonine 14 and tyrosine 15, activating phosphorylation on threonine 161, or all of the above. So CDK is of course being regulated by cyclin association by activating and inhibitory phosphorylations, as have been mentioned. And yeah, therefore the correct option is all of the above, or option 4. Ubiquitin is covalently conjugated to protein targets at which of the following amino acid residues? So we have histidine, arginine, lysine, or glutamine. The correct option in this case again is lysine, and lysine is often found to be quite prone or quite enriched in areas where ubiquitination needs to occur. Which of the following genes may complement the phenotype of Saccharomyces pombe CDC2 mutants? Human CDK1, human cyclin A, human cyclin E, or human ATM. So among these, the CDC2 mutant phenotype or the human homologue of the Saccharomyces pombe CDC2 is the human CDK1. Therefore, the correct option is human CDK1, which is option A. So if anyone has any doubts regarding these topics, any of the cell cycle topics as well, you can bring it up. And anything you wish to discuss also, again, please do interrupt me at any point of time if that is required. Okay. As part of week 3, we had majorly looked at ubiquitin proteasome system and how S phase is being how the S phase is being looked at. I think there was some rearrangement from that point of time, from 2021 till the current syllabus. But overall, you'll find similar questions for the same. So either way, it should be quite useful for you in the long run. Okay. So the first question is about ubiquitin attachment. So if you remember the way in which ubiquitination was being carried out, something we had discussed, that the way in which E1, E2, and E3 enzymes, after they have detected that a certain protein needs to be degraded from the cell at a particular point of time, they go and first carry out monoubiquitination, add more and more ubiquitin molecules, create a polyubiquitin chain, which is then recognized by the proteasomal system, and then it is taken forward for degradation. So just a second. Yeah. So this attachment of ubiquitin to the lysine amino acid side chain of these proteins that are undergoing degradation occurs at a particular position. It occurs for only one of the lysines, or usually occurs at one of the lysines, and that is the epsilon lysine. So that is the correct answer in this question. Just in case this was something you were not familiar with, you can refer to a any research paper or any online blog that will give you an idea about how proteins undergo ubiquitination and what are the molecular machineries that are responsible for smooth regulation and smooth execution of the same. Which of the following factors is not required for the formation of the pre-replicative complex on the origin? So, if you remember, we had discussed the different substeps, one of which was the prior assembly of the pre-replicative complex at the ORC sequence or at the origin. So we have been asked which of the following is not required for this purpose. Origin recognition complex, or the ORC, of course, is required. It binds to these sites. And CDC6 as well as CDT1 are also crucial. On the other hand, phosphorylated MCM proteins, they are not required for pre-RC or pre-replicative complex formation. Instead, their job is more towards the opening up of DNA or the unwinding of DNA so that the replication machinery can access the same. So therefore, option 3 is the incorrect, is not required in context of pre-replicative complex or pre-RC assembly. Then we've been asked which of the following is present in the proteins of ubiquitin-like protein family. A 5-stranded beta sheet, a short helix, which is a 3-10 helix, and also an alpha helix. The correct answer is all of the above, and therefore option 4 would be correct in this case. Pick the odd one out from the given options. The U box, the ring domain, and the HECT box, or rather HECT domain, as it is more commonly known. So among these, the odd one out is the destruction box, which is option 4, while the others are not particularly relevant in this regard. I don't think it was directly discussed in our live interaction sessions, but maybe this would be a helpful question for you just to keep in your mind. Then we come to question 5. Which of the following has been implicated in the regulation of histone stability for controlling gene expression? So, when we talk about the stability of histones, then a different concept will come in. Let us look at the options first. Whether it is the heterotypic ubiquitin chain or the branched ubiquitin chain, APCC, K1148 ubiquitin chains, all of the above, or whether only answer D or answer C and D. So the correct option is all of the above, because both heterotypic ubiquitin chains, branched ubiquitin chains, and APCC, as well as K1148, all of these have been implicated in controlling gene expression. Uh, Select the odd one out from the given options. From these, we can see THIS is the incorrect option. Ubiquitin, Sumo1, and NetD8 are not. Ubiquitin and Sumo1 are, of course, we know they are post-transcriptional modifiers. Therefore, the correct option in this regard will also be THIS. I think this might not also be something we have discussed much extensively. So if it is unfamiliar to you, you can skip it for the time being. We will shift our attention more towards the questions that are relevant based on the current syllabus. So we were asked, in which phase of the cell cycle does the licensing of replication origin occur? This is something we have discussed in our replication classes, that licensing occurs in advance. Therefore, it is actually necessary to make sure that all of the origins they fire, and they fire in a manner that it is well regulated, and it ensures that there is no misfiring of any replication origin during one cell cycle. So this licensing step is being carried out prior to initiation of DNA replication, and therefore it is executed during the late M or the G1 phase. Then I think it got stuck. Yeah. Now it's moving. So basically, what we see here is that since replication is executed during the S phase, and like we just discussed, the order in which the cell cycle phases are arranged is such that there is G1, S, G2, and M. Therefore, prior to the S phase, the cell cycle clock is stagnant within the late M and G1 phase, and therefore that is when the licensing of replication origins or ORIs takes place. In question 8, we've been asked which of the following statements is incorrect for S phase of the cell cycle. S phase is known as the synthesis phase of the cell cycle. True. The entry of a cell into the S phase is highly regulated. That is also true. Once a cell passes through the G1 restriction point or the start point, it gets committed to go through S phase. That is also true. That is why this particular position is called as the start point or the restriction point, because it is considered to be the point of no return. That once a cell has duplicated its genome and prepared itself completely for the upcoming division and subsequently for the process of nuclear division as well during mitosis, since it has carried out the preparations which are necessary for it. So once it commits for proceeding through the cell cycle, it will go through it. And finally, we have been asked whether the removal of external stimuli induces entry into S phase, causes the S phase cells to go back into the G1 phase. So if we like we just talked about the cell fusion experiments, that there is a directionality that is being maintained through the cell cycle. Once a cell has committed for entry into the S phase, it even if you remove the external stimuli that pushed it there initially, these S cells will not be S phase cells will not be able to go back into the G1 phase. So therefore, that is the correct option, option 4, because this statement is untrue and this statement is not feasible. External growth factors induce the synthesis of D-type cyclin in mammalian cells, and this cyclin is important for which of the following? The cells to cross the restriction point, for the inactivation of ORC, for the inactivation of E2F, or none of the above. So for cyclin D synthesis, you have external growth factors in place, and this cyclin is important in order to allow cells to cross the restriction point and enter into the S phase and commit to the process of cell division and the cell cycle. So therefore, that is the correct option for the cells to cross the restriction point. And finally, we were asked what is the subunit which is responsible for catalysis in the proteasome, the 26S proteasome, and the correct answer in this case is the 20S subunit. If you remember, we had the 19S top and bottom, and the central hollow pipe-like region, which is identified as the 20S subunit. So yeah, that completes the topics for week 3 as well. Again, let me iterate if there are any doubts. I'm asking this at the end of questions for each chapter. I think that will help in brushing you up about the topics that were there, and will give you a little better idea about how to tackle questions like this as well. Okay, so then we for just let yeah. Next, we were asked about the origin of E. coli in the this week basically covered the topics of DNA replication, the centrosomes, how they're arranged, what is their role in disease, and how they are developing over time or evolving over time. So first question is about the Ori-C arrangement. In case of E. coli, we have 13 and 9 mers, if you remember. The question is how many of each of them. Then there are 3 13 mers and there are 5 9 mers. Altogether, they create what is identified as the origin of replication for E. coli cells. Uh, Second, we are asked that the processivity of the DNA polymerase in the Pol3 holoenzyme is increased by which of these following? So DNA Pol3 we expect in the Hall enzyme to be quite effective, but to increase its processivity, which means that once the Pol3 holoenzyme has bound onto this onto this, then after that it should stay and it should persist, and it should not fall off during the process of replication. So this processivity is increased or is facilitated by the beta clamps, which, if you remember, they encircled DNA and they held DNA in the correct manner in order for the process of replication to be proceeded. And therefore, from this, you get an idea about how processivity and particularly the expansion of energy is very, very relevant to cells, and they will try to maximize the output that can be obtained with the minimal investment in exchange for the same. So the correct answer to that question is beta subunit. Then in question 3, I think, yeah. RNA primers in the lagging strand. So we had seen how there's a difference between the leading and the lagging strand synthesis. What are uh, the structural differences, the way in which the process is moving, the time that is taken, and the efficiency at which it is carried out. So RNA prime, just a second. Yeah, RNA primers that are initially added in order to provide a 3' OH for the enzyme to do its job. So this removal after synthesis has been carried out, these RNA primers have to be removed and then they have to be ligated once again with each other. So the strands that have been synthesized in place, they have to be stitched together to make one continuous fabric. So this digestion can be carried out by the DNA polymerase 1 enzyme, which does have this digestive capacity or this removal capacity. So that is the correct option for question 3. Then come centrioles. How many triplets of microtubules are actually present there? So this is again a very specific question, but if you're being asked about the composition of centrioles, then the number of microtubules that are operating in that regard are 9 in number. So there are 9 triplets or a much larger uh, number of centrioles, like the number of microtubules that are forming that particular centriole. And if you remember, we had talked about how these two centrioles, they are oriented in a perpendicular manner with each other, and they're surrounded by the pericentriolar matrix in order to form the cloud-like structure that we see, where the microtubules, they attach towards the cell boundary and they assist in the movement or the microtubule depolymerization, thereby allowing in the stretching of the cell and therefore eventually allowing movement or migration during anaphase. Then come in which phase of the cell cycle does centrosome duplication actually take place? So centrosome duplication, or rather any genetic duplication that we have talked about, all of that is completed during the S phase, which is the synthesis phase in this case, and therefore that will be the correct option for any duplication in this regard. The de novo centriole formation is associated with which of the following processes? So flagella formation, cell cycle, cell division, or cytokinesis. So if you remember, the way in which centriole formation is so connected to the movement of certain appendages within cells, in particular for flagella and cilia, then that would help you in understanding the way in which centriole formation is relevant in this context. Now, they do allow in the process of flagella formation, but and it is a de novo process. On the other hand, when we talk about cell division and the generation of these multiple copies or more than one copy of each centriole, then that's not a de novo process. It is a result of duplication that has occurred. Which of the following kinases induces both canonical and de novo centriole formation? So that is, I don't think we have discussed this, but for your own information, you can keep this in mind. It would be polo-like kinase 4. In question 8, we have been asked that DNA replication in E. coli and other prokaryotes as well as eukaryotes occurs in a semi-conservative manner, which implies which of the following. So I'm not reading out the options because they're quite redundant in nature. But basically, what happens is that you have a double helix, DNA double helix, and because of the semi-conservative nature of this replication, first those two strands, those two complementary strands, would separate from each other, and taking those individual strands as the template, as the parental template, a new complementary strand is synthesized, whereby the polymerase takes that as the template and introduces or incorporates the complementary nucleotide with respect to it, and that is carried out based on Chargaff's rules, like we just discussed. So that would be the correct option in this case, and that is option 3. I hope this is clear. It was a direct question by itself. Um, then coming to question 9, the defect in which the following proteins will lead to no replication fork formation is which of these following? So if no replication fork needs to be, if no replication fork is formed, then that means the very assembly or the formation of the pre-replicative complex and eventually beginning of or the initiating part of DNA replication is getting hampered, and that can be because of a defect or a mutation in the initiator protein INR, or otherwise also known as the DNA A protein, which is option 3 for question number 9. And finally, we have in question 10, the 1000 base pair long double-stranded DNA molecule contains 30% adenine. Determine the number of guanine bases. So I hope you're all already familiar with how you need to tackle these questions. Once again, the basic idea is to take Chargaff's rules into consideration. And like we said, that if your double-stranded DNA molecule, it has 30% adenine, then what is the total number of adenine residues? That's the first thing that you'll calculate. So 30% of 1000 will give you 300 adenine residues, right? It is 30 upon 100 into 1000. So you will get 300 bases or 300 adenine nucleotides that are present within this double-stranded DNA molecule. Now, from there, since we know based on Chargaff's rules that since that is the number of bases that are also like the number of adenine residues would be equal to the number of um, thymine as well, or A would be equal to T. Therefore, there are 300 adenine and there are 300 thymidine also present. Again, since G and C are also supposed to be equal in number, the remaining base pairs, I've been saying residues, sorry, that's a error on my part, they're all base pairs. But the remaining base pairs, which is 400 bases, are individually being occupied by this particular. Just a second, I think I am calculating something wrong. Okay, I think, okay, yeah, okay. I calculated it incorrectly. So if it is a 1000 base pair long double-stranded DNA molecule, then the total number of bases that are present within that DNA molecule would be 2000 bases. So for that, 30% adenine would include 600 adenine base pairs and or 600 adenine bases, and also in equal number you would have 600 thymine as well. So therefore, the remaining residues, which are 800, from those 800, half of them would be guanine and the remaining would be cytosine. Since we have been asked what is the number of guanine bases, therefore the answer to this would be 400 guanine residues, which is the correct option. Keep in mind that this question, while it was relatively direct, I approached it in an incorrect manner. Please don't make that mistake. Make sure that if it is the length of the DNA that is being mentioned, then that implies that if it is a double-stranded molecule, the total number of bases which would be present in that fragment would be double. Now, this may also so happen that the question might be a little more complicated because if you read the instructions, you would see that the exam can have a mix and match of different types of question. So you can have a essay kind of a question where they're starting off with a double-stranded molecule. They might be denaturing it at some point of time, or they might be carrying out some sort of ligation, or multiple processes can occur. So make sure that you keep a track of the number of bases at each step and then calculate accordingly. Otherwise, if it's a direct question like this, the answer is certainly easy to figure out. So yeah, so this actually indicates that you can get a few very basic numerical like questions as well, which should not be very difficult for any of you to tackle. But you can have a few questions in practice if that is more, helpful for you. So that was for the week 4. For week 5, there was mitosis and DNA replication as well. So yeah, I think this was a different arrangement. Okay, never mind. Okay. Uh, don't take the weeks into consideration here. The arrangement of syllabus was different back then. Just focus on the questions for your own understanding. These are PYQs from NEPTIL assignments only. So it should be more helpful for you to formulate the way in which you study or the way in which you approach questions in the long run. So yeah, here we are coming back to replication itself. So the first question is asking about the DNA polymerase enzymes that are involved in eukaryotic DNA replication. That which of the following statements are incorrect? That Paul delta synthesizes the lagging strand. That's true. Paul epsilon appears to yeah, Paul epsilon appears to synthesize the leading strand. That is also true. If you remember the way in which the assembly of Paul alpha and Paul delta, alpha, and Paul epsilon occurs, it is different. Paul alpha is majorly involved in the primase-like activity, which is why it brings about or it synthesizes the RNA primer. Whereas delta and epsilon are elongating polymerases, and among them, Paul epsilon is said to synthesize the leading strand, while Paul delta takes up the lagging strand. So all three of them are correct, and therefore none of them are incorrect, and the correct option is therefore none of the above. Then, in question 2, we've been asked that the early prophase of diploid cell has dash set of chromosomes. Telophase of a diploid cell has dash sets of chromosomes. First of all, you need to keep in mind that the question is asking for sets of chromosomes, not for the number of chromosomes. Your answer will differ accordingly. So when you have a diploid cell, initially before it has undergone nuclear decondensation and all of that, in the initial stages in early prophase where just chromatin has condensed and it has formed into this sort of structure, more organized structure. At that point of time you would have two sets of chromosomes. For the diploid cell, two sets would be present. On the other hand, the telophase of a diploid cell, how many will it have? So we have after this process of division has taken place and you have a cell that is about to undergo cytokinesis under such circumstances, that would also have two sets of chromosomes. So just give me a second. I think there might be an error here as well. Yeah. So I think this answer has been might have been incorrectly pointed out because um, it has not actually been mentioned whether the process is meiosis or mitosis. What exactly is taking place? If we consider a diploid cell, that is a diploid cell at the beginning of cell division and is a diploid cell at the end of division as well, then we have to answer accordingly. That if the ploidy of the cell is being dictated, then that means if it is a diploid cell at any stage, it means that the number of chromosomes that are present are 2 in number. So simply based on this information at hand, you can answer that it is 2 comma 2, that both in early prophase as well as in telophase, these cells would have two sets of chromosomes. However, if the process that the cell is undergoing or the type of cell division that the cell is undergoing is being mentioned, then you have to keep in mind that the number can vary depending on whether it is mitosis or it is meiosis. So you need to answer accordingly and very carefully as well. So this is why what this basically shows you is that you need to answer like you need to answer carefully, but you also need to analyze the question. You need to read it, look for cues that are being mentioned, and then tackle the question. Like I also took a split second to make sure that I'm answering it correctly because nothing in specific has been mentioned here, whether it is mitosis, meiosis, what type of cell it is. Nothing has been mentioned. It's just the ploidy. Based on the ploidy, your answer would be 2 comma 2. Then in question 3, the absence of telomerase activity in a cell, it results in which of these following? So, yeah. So, for telomerase activity, the reason why telomerase at all is present within the cell is for the maintenance of the ends of the chromosome, right? So, if there is an absence of telomerase or it is not present within the cell, then that would lead to progressive shortening of the chromosome ends as the cell would divide, and it would limit the number of somatic cell divisions that the cell can undergo, because beyond a point of time, what we'll see is that the telomeres would become so short that such a cell would not be allowed to or permitted to divide. So that is the correct option. That is option 1. Which protein can catalyze the formation of phosphodiester bonds? So for DNA ligase to come in, like for DNA ligase to perform its job, it needs to have an available 3' OH and a 5' phosphate, and it basically links those two and creates the bond that we very commonly know as the phosphodiester bond or the DNA backbone. So the protein which carries out, or the enzyme which carries out this process in particular, is the DNA ligase. Um, then during DNA replication, single-stranded DNA is kept from re-annealing due to the function of which of the following? DNA helicase, topoisomerase, RPA, or histones. So we had discussed the several different participants that come in during the process of replication. Among them, helicase, of course, opens up DNA. Topoisomerase is for bringing about some change in the torsion of DNA, which means the coiling or supercoiling of DNA is being relaxed in response to the opening up of DNA that is being carried out by the helicases. Histones are, of course, they have a whole other function. They are involved in the correct epigenetic, not epigenetic modification, but rather in the organization, genome organization or chromatin organization in the proper manner. Therefore, the single-stranded DNA binding protein, which is very crucial, which prevents re-annealing, is being bound by RPA, replication protein A, that binds to single-stranded regions of DNA which are undergoing replication or within the replication fork, and they are maintained in that state until the process of replication has been completed for that area. uh, the pre-RC or the pre-replicative complex is formed, we already discussed this, is formed during which phase of the cell cycle? So prior to the S phase or the synthesis phase, the pre-RC would be formed, and that is during the G1 phase. So that's the correct option. Um, then which protein helps in the tethering of DNA polymerase to the DNA? So if you remember that there is a complex that actually carries out this sort of loading, or this it provides this scaffold for other proteins to assemble and execute their activity. So in context of DNA polymerase, this is being regulated by PCNA, or proliferating cell nuclear antigen. So PCNA helps in the tethering of DNA polymerase to DNA. Then in question 8, we have been asked that the catalytic part of the telomerase enzyme is or possesses which of the following activities? So just a sec. Yeah. So basically, it is asking us about the way in which the telomerase enzyme works, and it is heavily dependent upon the reverse transcription activity, because it carries with itself an RNA primer, and with respect to that RNA primer, it synthesizes a complementary DNA sequence, and that is actually what gets added onto the telomere ends. So the reverse transcriptase activity, which is a RNA dependent DNA polymerase activity, is the correct answer for this question. So if you get a question like this, either of the two answers would be correct. Either you can write it as reverse, you can select the one that says that it is reverse transcriptase activity, or you can go for the one that talks about the RNA dependent DNA polymerase activity. That is, it takes RNA as the template and it synthesizes DNA in response. The site of attachment of the spindle fibers to the sister chromatids during mitosis is which of the following? The centriole, the telomere, the kinetochore, or the centrosome. So spindle fibers attach to the sister chromatids of a homologous of a chromosome during the mitosis at the kinetochore. So kinetochores are actually considered to be the site of attachment of microtubules. They are present near to the constriction site of that chromosome, wherever it might be present. And finally, which of the following events is not required for chromosome segregation during anaphase? First option is about the activation of APCC. That is, of course, correct. The degradation of securin, that is required because securin gets degraded, it releases separase, and then separase can execute its activity. The cleavage of cohesin, it is absolutely required because it is necessary for allowing those two sister chromatids to separate from each other and to no longer be held by these cohesive forces and instead be allowed to migrate in opposite directions and form two daughter cells from the initial parental cell. The incorrect option, or the option which says which indicates the event that is not a prerequisite for chromosome segregation, is the inactivation of separase. You actually don't want that. You want separase to be activated, and it is activated in a very controlled manner to execute its job. So yeah, that, just a second. Yeah. Yeah. So that completed the week 5 questions for DNA replication. Just let me zoom in a little. Yeah. Okay, so here there were topics like cytokinesis, aging, senescence, apoptosis part 1, apoptosis part 2, and meiosis. We have covered in the tenure of our course according to the plan or the schedule that has been proposed for this year. We have done this in a slightly different manner, but effectively all of the topics that get covered are the same at the end of the day. So the questions that were raised in this regard were firstly about which of the following is not a hallmark of aging. So there would be loss of proteostasis, that is protein stability or protein maintenance would be affected. Genome instability occurs with age. Telomere lengthening is something that will never occur. In fact, the telomere can shorten and does shorten with increasing age, stress, and a number of other factors. It is simply put, it is one of the reasons why people die, because with age, all of these hallmarks are observed and all of these changes take place, which eventually compromises the ability of an individual to survive indefinitely. Of course, mitochondrial dysfunction is also a hallmark of aging. Therefore, the correct option in this regard would be telomere lengthening. Then, second question is about senescence, and senescence is accompanied by first the decreased reactive oxygen species or ROS, the increase in apoptosis, cell cycle arrest, or inactivation of CDK inhibitors. If you remember, we had defined senescence, in fact, also in this manner, that it is a state of arrested cell division or arrested cell cycle, and it is also considered to be a hallmark of aging. So taking this in mind, the correct option is cell cycle arrest, because once a cell has decided that it needs to enter into senescence, then there would be an arrest of the cell cycle. In budding yeast, the cell division site is marked in which phase of the cell cycle? So if we talked about the way in which budding yeast actually works, and I don't think we have discussed it in this way directly. It might have been mentioned during the process of cell division in eukaryotes, taking yeast as examples. So the point at which cell division will occur is marked or is decided during the late G1 phase, because right after that we have the S phase and then G2 and M phase. So G1 is the time where the site of cell division, or the point from which the budding will occur, and the new daughter bud or younger bud will be pinched off. So that occurs during the late G1. Um, what are the components of the contractile ring in cytokinesis? Again, this is a very direct question. There is F-actin and myosin 2, which is the correct option. I'll not unnecessarily go through all of them. So these two proteins enrich the contractile ring, and that is an extremely crucial event in order to generate sufficient forces, sufficient mechanical force to allow the separation of the nucleus and finally of the cytoplasm as well, completing this step of cytokinesis. So the correct option is F-actin and myosin 2. MEN or MEN, otherwise known as the mitotic exit network, consists of A. Protein phosphatases like CDC14 phosphatase, kinases like CDC5, 15, DBF2, DBF20, GTP binding proteins like TEM1, LTE1, and BFA1 BUB2 complex. So the MEN, again, this I don't think I elucidated a lot on it during the live interaction session, but it is certainly there in sufficient details in the video lectures. So the correct option based on the mitotic exit network or MEN are all of the following, and that is the correct option in this case. What will be the effect of phosphatase inhibitors on nuclear envelope reassembly in telophase? In that case, if there is a phosphatase inhibitor that is being used, then in such cases, such as said, um, the assembly of the uh, nucleus basically, or the nuclear envelope will be stopped. So if phosphatase inhibitors that act upon the will go and act upon the nuclear envelope, then its reassembly would be prohibited or will not be allowed. And this particularly occurs during the telophase step after all the previous genetic material has been segregated from the mother to the daughter cell. Which of the following statements is not correct about apoptosis? Apoptosis of cells, it induces inflammatory reaction. It causes shrinkage of cells. Apoptotic cells, they show nuclear condensation and the fragment formation or fragmentation or none of the above. So we have been asked to pick the statement that is incorrect or false about apoptosis, and that is the induction of inflammation, because that is the key difference that uh, even in layman terms is understood by everybody. um, that the process of necrosis is uncontrolled in nature, and the process of necrosis is accompanied by very extensive inflammation, swelling, release of cellular compartments into the surroundings, and so on and so forth. But apoptosis is carried out in a very, very controlled manner, and therefore that statement is not correct. Caspases belong to which class? Very simply put, they are cysteine aspartases, which means they have a cysteine on them, but I mean they have a cysteine at the um, what is it called, in their active site, but their cleavage or their identification and a subsequent proteolytic cleavage occurs after the aspartic acid residue. So they are otherwise also known as cysteine proteases because they carry a cysteine within the protein, and that allows it to execute its own catalytic activity. And in the next two questions, we've been asked which of the following is true about cytochrome C release from the mitochondria. So we talked about the intrinsic and extrinsic pathways for the way in which programmed cell death is being regulated, and we are being asked which of these is actually true about that process. A, that it occurs during mitosis. Certainly not. It activates P53. No. Its release is not regulated by the BCL2 family of proteins. That is also not true. They are proapoptotic in nature. They do carry out cytochrome C release, and therefore the incorrect wait. No, BCL2 family is not regulating it. I said it incorrectly. BCL2 will not allow apoptosis. It is anti-apoptotic in nature. Sorry, I was thinking about back. But yeah, the correct option in this case is that cytochrome C release is eventually going to trigger caspase activation in cells. And in fact, this is the way in which the intrinsic apoptotic pathway is also triggered, initiated, and maintained. That cytochrome C release along with calcium eventually triggers the activation of caspases, then the cascade will set up, and then eventually they will merge into the same direction and the cells will die. Finally, we've been asked which of the following statements is true. Then, apoptotic, apoptotic death is not executed by proteases. Death induced by FAS or TNF causes the activation of caspase 8 via the death receptor complex. That is true. Okay, let's just go it option by option. The first one is apoptotic death is not executed by proteases. This is not true. It is very, very heavily dependent upon caspases, which are proteases in nature. Second statement appears to be correct. Increased apoptosis is a hallmark of many cancers. This is absolutely false. In case of cancers, we have seen that evasion of apoptosis or bypassing cell death or resisting cell death is a key hallmark of most cancers, that they keep on trying to proliferate and increase a number despite carrying all sorts of mutations, and therefore apoptosis is reduced, removed, or completely eliminated unless it is triggered by some specific signal. And finally, the inhibitor of apoptosis proteins are endogenous inhibitors of BCL2. So again, this is also not true. So the correct option is option 2, that FAS or TNF, tumor necrosis factor mediated or induced death, causes caspase activation via the death receptor complex. So we had seen how FAS or TNF, they bind to their receptor, then that receptor for dimerizes, it forms the death receptor complex, and then it eventually triggers the programmed cell death process. So that is the correct option, option 2. Um, then, yeah, as part of week 7 in this course, meiosis, nuclear organization, chromatin organization, SMC proteins, DNA imaging, cohesion complex, all of this has been covered. So to go through the questions in that regard, let me just zoom it a little bit. Yeah. So, which stage in meiosis is characterized by the occurrence of crossing over between homologous chromosomes? So, we had discussed the substages, the LZPD squared, which was leptotene, zygotene, pachytene, poltene and diakinesis, diplotene and diakinesis. So, LZPD squared were the different substeps or the subcategories of prophase. So, in case of meiotic prophase, the crossing over event, it occurs during pachytene. You can very distinctly see the formation of bivalent, and you can see these chiasmata, which are the points of crossing over. Second, non-disjunction of chromosomes occurs due to which of the following reasons? Premature sister chromatid segregation, homolog pairing is aberrant, missegregation of homologous chromosomes, or all of the above. So non-disjunction can occur because of any of the following. So all of the above is the correct option, that either the sister chromatids separate before the microtubules have attached properly, the way in which the homologs have paired, it is not correct, it is abnormal or incorrect, and then we can see that there is there could be missegregation, that despite correct attachment, it might just so happen that instead of the two sister chromatids migrating in opposite directions to either microtubules, they both migrate in the same direction, and the other daughter cell lacks a copy of that particular chromosome. So that is also a that's also a possible mechanism via which you can often find non-disjunction to take place. Hmm. The chromosomal number is reduced to half during which of these? So it is the options here are meiosis 1, meiosis 2, mitosis, or binary fission. So reductional division or meiosis is the process where chromosome number reduces to half, and in particular during meiosis 1 this takes place, whereas during meiosis 2 it is much alike to the mitotic substeps, and in mitosis and in meiosis 2 we see equational division, that the initial mother cell and the daughter cells, they have an equal number of chromosomes. Then coming to question 4, the correct sequential order of stages occurring in the prophase 1 of meiosis are, like I just said, LZPD square or leptotene, zygotene, pachytene, diplotene, and diakinesis. Um, in question 5, We've been asked to pick out the correct statement regarding the role of condensins in chromosome structure maintenance. So, firstly, the complex tethers DNA regions between sister chromatids in order to generate loops that compact DNA. Without condensation, the leading chromosomes are cleaved by cell division. Not true. The complex tethers DNA regions within chromatids in order to generate loops that compact DNA. Now, this might come to mind that what is the difference between the first and the third option? If you read it very carefully, you'll see that the first option is talking about the regions between sister chromatids. So this cannot happen, and it is actually quite detrimental as well, because eventually the two sister chromatids under normal circumstances should separate and segregate independently into two daughter cells. And in order to ensure that, it must also be ensured that the way just yeah the way in which this looping is being carried out, that is only within one sister chromatid or within the same sister chromatid, and therefore that is the correct option. Option 3 is or option C in this case is correct. In option D, we've been told that without condensation, lagging chromosomes would be cleaved off or removed during cell division. So that is also correct. And yeah, therefore both C and D would be the correct options in this case. How many condensin molecules act upon the loop extrusion of DNA according to the double tethered DNA assay? Remember, this was directly, it was a part of one of the external video lectures. And here what they saw was that in a loop extrusion of DNA, the number of condensins that actually bind are 1. Then pick out the wrong statement about the SMC protein complexes. They're long and flexible. They're conserved in all life forms except in prokaryotes. This is not true. Then the SMC proteins have coiled regions. That is certainly true. We have discussed the structure extensively. There are coiled regions which are being interrupted by those hinge regions which are quite flexible in nature, and that is actually necessary for proper and strong binding with the DNA sequence where it is supposed to localize. And the SMC complexes, they have an ATPase domain. That is obviously also true. They need to carry out their energy dependent activities. Therefore, the only wrong statement is option 2, that it is conserved. SMC proteins are conserved in all life forms except prokaryotes. That's not a necessary criteria, and therefore that is the wrong statement. Pick the correct order of steps in performing the 3C technique. This was even part of one of the assignments that we had this year. So I'll get to the option directly, that you cross-link, restriction, digest chromatin, ligate, purify and then paired end reads can be sequenced in order to get an idea about the region. So yeah, that was question 8. In question 9, we've been asked which of the following has no effect on gene expression. Um, the chromosome position certainly has an effect upon gene expression, where it is present, whether that region is heterochromatinized, how accessible is it to the transcriptional machinery, how often does it undergo mutations. All of that can affect that the expression of that particular gene. The subcellular localization also has an impact, whether uh, uh, it's a nuclear protein, it's a cytoplasmic protein, it's a tissue specific protein. All of that can also make a lot of difference. Histone modification also matters. So technically, all three of them have an effect upon gene expression. So the correct option would be option 4. Finally, which of the following are synthesized in the nucleolus? So you have mRNA, rRNA, heat shock proteins, or all of the above. So yeah, I hope everybody is familiar with it. The compartments of the nucleus and how they are uh, utilized. So the nucleolus and the nucleoplasm, how they're utilized, what is produced in that region, why is it not produced elsewhere, etc. This can all be interesting areas of studies, and you can look into it if that makes sense to you as well. So the correct option in this case is that within the nucleolus, um yeah, within the nucleolus, the rRNA or the ribosomal RNA production is carried out. That will help in assembly of the macromolecular ribonucleoprotein complex with the catalytic activity. And finally, we'll go through some questions, which should end the discussion for today, unless you have any doubts or any inputs. Okay, was there an issue? I think I got disconnected briefly. Yeah. Um, yeah. So for the last few questions, what are all the criteria that were checked for the validation of the gene edited cell? So, precise tagging, stable expression of the, this was actually part of the extra video lecture about stem cells and how you can, use different technologies to study them, etc. So, for a cell that has undergone gene editing, so the criteria that is necessary for validation of the same would be actually all of the options that are given here. It needs to be tagged precisely. That protein needs to be tagged so that it can be monitored or its localization can be determined. Its stable expression must be carried out. And of course, because we are tagging it properly, we expect that localization to also be visible and easy to determine. So the correct option is option 4, that is all of the above. Second, in the meiosis recombination pathway, DSPs or double strand breaks are induced by HO endonuclease, SPO11P, SPO13P, or REC8P. So, this was something we discussed extensively in the live interaction session as well. So, you will find presentations on this for your reference if you need it. So, DSP generation is very, very heavily dependent upon the SPO11. Endonuclease had a different role to play. We discussed it more recently in the week 8 discussion, where we saw that HO endonuclease has a role to play in the yeast mating type switching. That is from the MAT A to MAT alpha or vice versa. There, the recombination is being carried out by that particular endonuclease. So do not get confused between the two. Um, this has a separate role. This has a whole other role for DSP induction for regular meiosis recombination that is taking place. SPO11 will do the job. Um, yeah. Then double strand breaks get repressed or are repressed in which of the following regions? The telomeres, the centromere proximal region, single copy coding regions, or both A and B. So, of course, the DSBs are not found in telomeres. They are also not found very close to the centromere because it's a very inaccessible position. But certainly in single copy coding regions, you will find there is a higher propensity for uh, double strand break formation. Therefore, the correct option would be both A and B, that is for both telomeric regions and centromere proximal regions. The meiosis specific variant of MCD1P, SCC1P in the cohesin complex is known as, I don't really have much to explain here. It's a direct recollection question. So some questions you will always get even in the exam as well. Some of them are just uh, memory based, how well you can remember the intricate details. That will give you an edge above everybody else who is also appearing for the exam. But yeah, it's not always necessary or not even feasible to remember every tiny detail. So if your concepts are clear, then you'd be able to answer correctly more than enough questions that will get you really good scores in the exam, if not the highest. So please make sure that you are answering correctly and you are answering in a way that you are confident about that particular answer. Otherwise, you can wait it out. So here, because it's a direct question, I'm not getting into details. It is REC8P, which is the meiosis specific variant for the same. Which of the following techniques can be used for studying protein-protein interactions? The GFP tagging of protein of interest, yeast to hybrid system, yeast monohybrid system, or plasmid shuffling. So it is actually Y2H. We also discussed this in the last interaction session on Sunday, how the yeast to hybrid system works, how it is it's one of the most remarkable, one of the most brilliant techniques for understanding protein-protein interactions. If you want, you can read more about these. There are a number of fluorescence associated techniques as well. For example, FRET, which depends upon the energy transfer from one fluoro labeled tag to another fluoro labeled tag to generate different color of light, and that can be easily detected. So you can look into it if it is interesting or relevant to you as well. Then a marker that is used for the selection of colonies by selecting for the loss of the marker whose expression essentially kills the cells is called which of these following. So we have kind of discussed almost all of them, but here you can go through the question a little more conceptually and it will help you arrive at the answer. So basically you are using some sort of a determinant or some sort of a marker where you want only bacterial colonies of a certain type to survive. For that, what are you doing? You are selecting for only those cells in whose case, if there is a loss of that marker, then such a marker will not how do I say that if that marker is expressed, then under those circumstances the cell will die. On the other hand, if that marker is absent, then that cell will persist. So it's an easy way of making sure that only the cells that have your desirable phenotype and have lost that particular marker, only those cells will survive. So this strategy, which is slightly indirect in nature, is for counter selection marker, and therefore you make sure that only cells which have lost the marker that you had initially used are allowed to survive, and then that is your that is your relevant population, let me put it that way. Which of the following can be used for plasmid shuffling while using the URA3 gene as a marker? I think it was there in the video lectures, but I don't think I discussed it myself. But yeah, it was mentioned in the video lectures directly. So the FOA, 5-FOA, it can be used for plasmid shuffling when you are using the URA3 gene as the marker. Then a recombination checkpoint operates during which phase of the meiotic cell cycle? Um, we had talked about previously, we talked about the checkpoint for entry into the S phase for a cell to go into division. But when we talk about the meiotic cell cycle, then within the cell cycle for recombination to be checked or to be monitored. The focus is on Pachytene stage of the prophase 1. So it is actually during the Pachytene stage, like we said previously, you would be able to see those bivalents or those chiasmata or sites where recombination has occurred, and then you would eventually be able to detect that yes, there has been crossing over and there has been recombination or exchange of information between these two regions, and therefore monitoring of that same is being carried out during the Pachytene stage, which is the correct option. Um, which of the following is not a phytochrome or not phytochrome, sorry, it's not a phytohormone? Auxine, cytokinin, ethylene or gibberellin. All four of them are phytohormones, and yeah, therefore the correct option is none of the above. And finally, for the last question, in budding yeast, the IME2 protein acts as which of the following? Meiosis specific transcription factor, meiosis specific phosphatase, meiosis specific inhibitor, or meiosis specific kinase. So IMA2 in budding yeast act as a kinase which is specific to the process of meiosis. So yeah, with this, yeah, I've come to an end of the questions that I wanted to share. Yeah, if there are any questions at this point, please go ahead and ask, because from my end, I'm done for the discussions for now. So whatever queries you may have, please go ahead with them.
Good evening, ma'am. Hello. Hello. Can you hear me?
Yeah, hi, I can hear you now. Yeah.
Yeah. Ma'am, actually, when I have uploaded this, hall ticket, something is mentioned that it will be a multiple choice as well as something we have to write. So kind
Yeah.
of questions also will come, MCQ as well as the writing questions, objective also, and
I
subjectives.
think somebody had asked this day before yesterday as well, in that session also. So if you don't even need to go to your hall tickets, I showed it that day as well. Just give me a minute. So if you just go to the announcement sections on the website itself, it was mentioned a few days ago that in the exam format, you will have all types of questions. You will get a mix of these multiple choice questions, fill in the blanks, essay type answers, etc. I don't think they will be that extensive that you need to write like paragraphs, but yeah, it could be a like a subjective question, basically, how you see. And fill in the blanks would be something that, we saw even now here in a couple of questions, that it might not necessarily be something to type, but it could be uh, multiple blanks that have been given, and you just need to figure out the answer for that. It could be in that format also.
Okay. And one more doubt I have, is there any negative marking for the wrong answer?
This I'm actually not sure. You can contact somebody from the team if there is a question, or you can ask. I don't think it's been mentioned here. Yeah, you can ask it on the discussion forum. I'm actually not aware of it. But I don't think there is a negative marking from what I recall from the previous years. But you can, just to be sure, you can ask somebody. It might come on your hall ticket as well. But yeah, to be honest, I'm not aware of it.
Actually, it's not mentioned anywhere. In
Okay.
instructions also, it is not mentioned. I don't, but still, I just have because if our answers are wrong and then if you are not sure, and you are
Yeah,
mentioning
so
that.
that will definitely be mentioned. I don't think it's here as well. It will definitely be mentioned at the time when you start your exam. Like if you're giving a computer-based test or even a pen paper-based test, then it is always mentioned what is like the mark for it, the positive marking for each question and the possible negative marking for it. So that you'll be made aware of before you attempt your questions. But right now, if you want to know, you can contact somebody from the team. They have a support team that is always working, so they will be able to get back to you. You can mail them also if it is, if you have access to any of their email IDs. And yeah, that should be good enough for you. And you should you anyway prepare in a way that you'll be able to answer questions as many as possible. But yeah, be careful if there is negative marking in case there is any. Be careful that you attempt your best. Do not compromise on the number of questions that you attempt. But yeah, be a little, wary before you. Be a little cautious and don't unnecessarily lose marks. But yeah, I'm not absolutely sure if there's negative marking or not. So I think you can wait it out or you can contact somebody from the team.
Okay, thank you, ma'am.
Okay.
Thank you, ma'am.
Thank you.
Yeah, okay. Anybody else has any doubts? Something related to the entire like the theme that we have discussed till now? Okay, if not, then thank you so much for joining. As part of your preparation, I would suggest you go through all of the slides that have been uploaded, not just by me, but by all the other TAs also. You would have access to the questions that they have formulated either on their own or any other PYQs that they might have. So go through those. If you have any doubts, since I don't think you have enough time to go back and go through everybody's video lectures, that will take up a lot of time. So for now, go through the slides, and if there's something that you want to be a little more clear about, you can go back to the video from there and see what they have explained in that regard. You can approach any of the TAs for any doubts that you might have. You can approach me as well on the discussion forum, and I'll try to get back to you as well. Um, Apart from that, if there are any exam conducting, like the way in which it will be conducted and the format, etc., if there are questions like that, uh, it would be better to go ahead and contact the team. They would give you a much clearer answer regarding this. And any doubts finally that you may have will obviously get resolved when you are appearing for the test. Yeah, besides that, go through the lectures well. If you want to browse through them quickly, you can look at the transcripts, because that if you are quicker at reading instead of going through the video in details, you can do that instead. You can quickly browse through the content that was discussed in that slide, so that you can get a quick recap of what has been discussed in 8 weeks. And yeah, you can use my slides as reference. If there's a particular topic you want to know more about, go to the reference book. I have mentioned, I don't think I've missed it anywhere. So I've mentioned the reference books that I've taken, whatever images or whatever concepts I've taken it from, everywhere. Even in some cases, there are certain papers that I also have cited. So yeah, that you can do. And anything that in the last moment that you need any terminology that you are unsure of, not absolutely sure of, you can just Google it, find the quickest way which will help you understand that concept and have a clear idea about it. So yeah, I think we will conclude our session with this. Once again, thank you so much for joining. All the best for your exam. I hope everybody does really, really well. And if there are any more queries, any more doubts, please go ahead and ask them on the discussion forum. We'll try to get back to it as soon as possible. And yeah, prepare well for your exams. All the best. And thank you so much for joining. Good night.
Thank you.